Welcome, and thank you for joining me here on Sleeping Researcher. This channel is dedicated to gentle science narration, designed to help you relax and drift peacefully to sleep. Today, we will explore the fascinating world of microbial ecosystems and their profound impact on life. Though invisible to the naked eye, these tiny communities of microorganisms shape the environment around us in ways both subtle and immense. From the soil beneath our feet to the depths of the oceans, microbes create delicate balances that sustain countless forms of life. As we journey through this quiet universe, we will uncover how these microscopic beings influence everything from nutrient cycles to human health. Understanding microbial ecosystems reveals the interconnectedness of life on Earth and reminds us of the intricate harmony within nature. If you enjoy this gentle exploration of science, please consider liking the video and subscribing to Sleeping Researcher. Your support helps us continue to share soothing scientific stories that calm the mind and nurture curiosity. Now, let us begin our calm voyage into the hidden world of microbes, where life thrives in the smallest of forms and quiet wonders await discovery. Imagine a world teeming with life so small that it escapes the naked eye, yet so vital that it forms the very foundation of existence on our planet. This hidden realm is made up of microbial ecosystems, intricate communities of bacteria, archaea, fungi, and other microscopic organisms that inhabit every corner of the Earth. From the depths of the oceans to the soil beneath our feet, and even within our own bodies, these microorganisms weave an invisible web that sustains life in countless ways. Their influence extends far beyond their size, shaping environments, driving essential biochemical cycles, and supporting the health of larger organisms. As we begin to explore this unseen universe, we uncovered the profound impact that microbial ecosystems have had and continue to have on the balance and vitality of life on Earth. Microbial ecosystems are remarkably diverse and dynamic, adapting to nearly every habitat imaginable. In the soil, microbes break down organic matter, recycling nutrients that nourish plants and, by extension, entire food webs. In aquatic environments, they regulate elemental cycles such as carbon and nitrogen, influencing climate patterns and the productivity of marine life. Even extreme environments like volcanic vents or frozen tundras host specialized microbial communities that thrive where few other organisms can survive. This adaptability highlights the resilience and versatility of microbes, whose metabolic activities are crucial for maintaining ecosystem functions. Their interactions within these microbial communities also foster cooperation and competition, creating complex networks that sustain ecological stability over time. Beyond their environmental roles, microbial ecosystems intimately connect with animal and human health. The human microbiome, for example, is a vast and diverse ecosystem residing within our bodies, particularly in the gut. These microbes assist in digestion, synthesize essential vitamins, and modulate immune responses. Disruptions in this delicate balance have been linked to various diseases, highlighting the importance of microbial harmony for well-being. Similarly, microbes influence agriculture by promoting plant growth and protecting crops from pathogens, reducing the need for chemical fertilizers and pesticides. Understanding these relationships opens new frontiers in medicine, agriculture, and environmental management, emphasizing that microbial ecosystems are not just background players, but active agents shaping our lives. As we delve deeper into the study of microbial ecosystems, we begin to appreciate the intricacy and interconnectedness of life at microscopic scales. These communities serve as both architects and caretakers of the biosphere, executing processes that maintain the flow of energy and matter, essential for all living beings. The invisible world of microbes invites us to reconsider our place within nature, reminding us that the smallest forms of life hold immense power to influence planetary health and sustainability. This exploration sets the stage for a deeper understanding of the mechanisms through which microbial ecosystems operate and their far-reaching consequences for life on Earth. Microbial ecosystems are intricate communities composed of countless microorganisms, including bacteria, archaea, fungi, viruses, and microscopic algae that coexist and interact within specific environments. These habitats can range from the soil beneath our feet to the depths of the ocean, from the human gut to the icy expanses of polar regions. Each microbial ecosystem is defined not only by the types of microbes present, but also by the physical and chemical conditions that shape their existence. Within these microscopic worlds, organisms engage in complex networks of cooperation, competition, and communication, forming dynamic systems that adapt and respond to changes in their surroundings. Understanding microbial ecosystems means recognizing the invisible yet vital threads that weave together the fabric of life on Earth. As we begin to explore these communities, it becomes clear that microbial ecosystems are not isolated pockets, but integral parts of larger ecological processes that sustain biodiversity and planetary function. At the heart of microbial ecosystems lies the principle of interdependence. Microbes frequently exchange nutrients and genetic information, creating symbiotic relationships that enhance survival and productivity. For example, in soil ecosystems, bacteria and fungi collaborate to decompose organic matter, releasing nutrients that nourish plants and other organisms. In aquatic environments, photosynthetic microbes convert sunlight into energy through photosynthesis, forming the base of food webs that support diverse marine life. These interactions are governed by chemical signals and environmental cues, enabling microbial populations to regulate their growth and activity efficiently. Moreover, microbial ecosystems demonstrate remarkable resilience, often recovering from disturbances by reorganizing their community structures. This adaptability is crucial in maintaining ecosystem stability and function, especially in a rapidly changing world. Through studying these processes, we gain insight into the fundamental roles microbes play in regulating Earth's biogeochemical cycles. Beyond their ecological functions, microbial ecosystems have profound implications for human health and industry. The human body itself hosts vast microbial communities, collectively known as the microbiome, which influence digestion, immunity, and even mental well-being. Disruptions in these delicate microbial balances can lead to disease, highlighting the importance of understanding microbial ecosystems within our own biology. In agriculture, harnessing beneficial microbes can improve soil fertility and crop yields, reducing the need for chemical fertilizers. Similarly, in biotechnology, microbial ecosystems inspire innovations ranging from waste treatment to the production of biofuels and pharmaceuticals. These applications illustrate how recognizing the complexity and potential of microbial ecosystems opens pathways toward more sustainable and harmonious interactions with the natural world. As we continue to uncover the layers of microbial life, we appreciate the profound impact these tiny organisms have far beyond their microscopic scale. The study of microbial ecosystems also challenges traditional perceptions of individuality and community in biology. Instead of viewing organisms as isolated entities, microbial ecosystems reveal a collective existence where survival depends on cooperation and shared resources. This perspective encourages a holistic understanding of life, where boundaries between species blur and genetic material flows freely through horizontal gene transfer. Such genetic exchanges allow microbial populations to acquire new traits rapidly, adapting to environmental pressures and emerging challenges. This fluidity in microbial communities contrasts with the more static views of macroorganisms, emphasizing the dynamic nature of life at microscopic levels. By embracing this interconnectedness, we open new avenues for exploring evolution, ecology, and the resilience of life itself. As we deepen our exploration of microbial ecosystems, we begin to unravel the mechanisms that sustain these complex communities. From nutrient cycling to energy flow, from symbiotic partnerships to environmental adaptation, microbes orchestrate processes that are essential for the health of ecosystems worldwide. These invisible architects shape the atmosphere, influence climate patterns, and support the food chains upon which all larger life depends. Their ubiquity and versatility underscore the profound truth that life thrives not only in what we see, but also in what remains unseen. This understanding lays the foundation for appreciating the broader impacts of microbial ecosystems, guiding us toward more informed stewardship of the natural world. The microbial world is a realm of astonishing diversity, where countless forms of life exist in unimaginable numbers and variations. From the tiniest bacteria to complex communities of archaea, fungi, and microscopic algae, microbes inhabit nearly every corner of the planet. Their adaptability is truly remarkable. They thrive in environments that would seem inhospitable to most other organisms. Whether in the frozen tundras, the scorching deserts, deep beneath the ocean floor, or within the human body itself, microbes have evolved to exploit diverse niches. This vast array of life forms not only reflects their resilience, but also their ability to innovate biologically, developing unique metabolic pathways and survival strategies. Just as forests and coral reefs are celebrated for their biodiversity, so too must we appreciate the microbial ecosystems which underpin the fabric of life on Earth. Microbial diversity is not merely a catalogue of species. It is a dynamic interplay of genetic variation and ecological function. Each microbe carries a set of genetic instructions that enable it to perform specific roles within its community. Some microbes specialize in breaking down organic matter, recycling nutrients that sustain plant and animal life. Others engage in symbiotic relationships, such as nitrogen-fixing bacteria that live in the roots of legumes, enriching soil fertility and supporting agriculture. In aquatic environments, photosynthetic microbes contribute significantly to oxygen production and carbon cycling, influencing global climate systems. This adaptability allows microbial communities to respond to environmental changes, maintaining ecosystem stability even under stress. Their ever-shifting populations and interactions embody a resilience that is foundational to life's continuity. The presence of microbes in every environment underscores their integral role in Earth's biosphere. Soil, often perceived as inert, is teeming with microbial life that drives decomposition and nutrient availability. These tiny organisms form complex networks, communicating chemically and exchanging genetic material, which enhances their collective ability to adapt. In the vast oceans, microbial mats and planktonic communities form the base of marine food webs, supporting fish, mammals, and ultimately human populations. Even extreme environments such as hydrothermal vents and acidic hot springs harbor specialized microbes that sustain unique ecosystems. Their existence in such varied habitats challenges our understanding of life's limits and expands the possibilities for where life might be found beyond our planet. This remarkable distribution and diversity invite a deeper appreciation of microbial life as a fundamental component of Earth's natural heritage. The adaptability of microbes also manifests in their ability to evolve rapidly in response to environmental pressures. Horizontal gene transfer, mutation, and rapid reproduction allow microbial populations to adjust to new challenges, such as antibiotic presence or shifts in temperature and pH. This agility is crucial in maintaining ecological balance, as microbes can quickly fill vacant niches or alter metabolic functions to sustain resource cycles. Their metabolic versatility ranges from utilizing sunlight to metabolizing inorganic compounds like sulfur or methane, revealing a spectrum of survival strategies that sustain ecosystems under diverse conditions. This capacity for adaptation not only ensures microbial survival, but also supports the resilience of the broader ecosystems they inhabit. Such intricate adaptability highlights the profound interconnectedness between microbial life and the environment. Exploring the astonishing variety and adaptability of microbes reveals a hidden world where life thrives in forms and functions that continuously surprise and inspire. Their omnipresence from soil depths to oceanic expanses is a testament to the intricate balance and interdependence of natural systems. As we continue to investigate these microscopic communities, we uncover new insights into their roles not only as individual organisms, but as crucial players in global processes. This expanding knowledge encourages a shift in perspective, inviting us to recognize the unseen microbial influences that shape every aspect of life and environment. Our journey into the diversity of microbial life thus opens pathways toward understanding the broader implications of these communities on planetary health. Microbes are far more than mere inhabitants of their environments. They are active architects that continuously shape and transform the very ecosystems they occupy. Through their metabolic activities and communal behaviors, microbes engineer their surroundings in ways that sustain life on multiple scales. One of the most striking examples of this is the formation of biofilms. Complex, structured communities of microbes embedded within a self-produced matrix of extracellular polymeric substances. These biofilms not only provide protection and stability to their microbial residents, but also alter the physical and chemical properties of surfaces, whether on submerged rocks in a stream or the roots of plants in soil. By creating such microenvironments, microbes influence nutrient availability, moisture retention, and even the colonization patterns of other organisms. This dynamic interplay between microbes and their habitat reveals an intricate feedback system where microbial life both adapts to and actively modifies its ecosystem. This perspective invites us to appreciate microbes as indispensable engineers whose unseen work underpins the health and resilience of many natural systems. Beyond structural modifications, microbes play pivotal roles in driving nutrient cycles that sustain ecosystems across the globe. Their biochemical pathways mediate transformations of elements such as carbon, nitrogen, sulfur, and phosphorus, facilitating the flow of energy and matter through food webs. For instance, nitrogen-fixing bacteria convert inert atmospheric nitrogen into forms usable by plants, thereby enriching soils and supporting terrestrial life. Similarly, microbial decomposers break down organic matter, releasing nutrients back into the environment and maintaining soil fertility. In aquatic systems, microbial communities regulate the cycling of carbon through processes like photosynthesis and respiration, influencing not only local food chains but also global climate patterns. These activities underscore microbes as essential agents in maintaining ecosystem productivity and stability. Their metabolic versatility and adaptability allow them to respond to environmental changes, demonstrating an ongoing, active engagement in shaping the conditions necessary for life. The influence of microbial ecosystems extends into symbiotic relationships that further illustrate their engineering capabilities. Within the roots of plants, mycorrhizal fungi form networks that enhance nutrient and water uptake, effectively expanding the physical reach and resilience of their hosts. In the animal kingdom, gut microbiomes assist in digestion, immunity, and even behavior, highlighting how microbial partners contribute to the health and survival of larger organisms. These symbioses often involve complex chemical signaling and resource exchange, reflecting a sophisticated level of ecological engineering at microscopic scales. Such interactions not only benefit individual organisms, but also influence community structure, biodiversity, and ecosystem function. Through these partnerships, microbes integrate themselves into the fabric of life, demonstrating their fundamental role as ecosystem engineers that enable and sustain diverse biological communities. This intricate connectivity between microbes and their hosts prompts us to look deeper into the interwoven networks that define living systems. Moreover, microbial ecosystems exhibit remarkable resilience and adaptability, qualities that are crucial in an ever-changing world. Their rapid growth rates, genetic diversity, and horizontal gene transfer capabilities allow microbial communities to adjust to environmental disturbances such as pollution, climate shifts, or habitat alteration. By modulating ecosystem processes such as nutrient cycling or organic matter decomposition, microbes can either buffer ecosystems against change or facilitate transitions to new states. In some cases, microbial activity can remediate contaminated environments, breaking down pollutants and restoring ecological balance. These functions highlight the potential for harnessing microbial engineering in environmental management and restoration efforts. As we deepen our understanding of microbial ecosystems, we begin to see how their natural capacities might be guided to enhance planetary health and sustainability. Such insights emphasize the active and dynamic roles microbes play, continually shaping the living world in profound ways. In contemplating the myriad ways microbes engineer their environments, we recognize a fundamental truth. Life itself is inseparable from the microbial processes that maintain and transform the biosphere. From the minute microhabitats within a drop of water to the vast interconnected networks beneath forest floors, microbial ecosystems form the groundwork upon which complex life depends. Their influence extends beyond the visible, orchestrating cycles and structures essential for the continuity of ecosystems. This awareness opens a window to new scientific frontiers, where exploring microbial interactions and their environmental impacts can illuminate pathways toward more harmonious coexistence with the natural world. As we prepare to delve further into the implications of these microbial forces, we turn our attention toward the diverse strategies microbes employ to thrive and impact life on Earth. Among the most profound examples of microbial influence on life are the intimate partnerships formed between microbes and plants. These alliances, often invisible to the naked eye, are vital to the growth and health of terrestrial ecosystems. One such relationship is the symbiosis between nitrogen-fixing bacteria and leguminous plants. In this remarkable partnership, bacteria residing within specialized root nodules convert atmospheric nitrogen, an abundant but unusable form for most plants, into ammonia, a form readily absorbed and utilized for growth. This biochemical exchange not only nourishes the host plant, but also enriches the surrounding soil, enhancing fertility without the need for synthetic fertilizers. Through this natural process, microbial symbionts act as living catalysts, transforming inert elements into life-sustaining nutrients, thereby underpinning the productivity of ecosystems, ranging from small garden patches to vast agricultural fields. Beyond nitrogen fixation, a diverse array of microbial symbionts contribute to plant vitality through multiple mechanisms. Mycorrhizal fungi, for example, weave an extensive subterranean network that extends the root system's reach, facilitating the absorption of water and essential minerals like phosphorus. In exchange for carbohydrates produced by the plant through photosynthesis, these fungi enhance the plant's resilience to environmental stresses such as drought, soil salinity, and pathogen invasion. This mutualistic exchange demonstrates a sophisticated level of cooperation where both partners derive benefits critical to survival. Furthermore, the presence of these microbial communities can influence plant hormone levels, modulating growth patterns and triggering systemic defenses. Such finely tuned interactions exemplify how microbial ecosystems function as integral components of plant life, shaping not only individual growth, but also the broader ecological balance. The influence of microbial symbiosis extends beyond individual plants to the composition and productivity of entire ecosystems. By enhancing nutrient cycling and improving soil structure, symbiotic microbes promote plant diversity and succession, which in turn supports a variety of animal life. In forest environments, for instance, mycorrhizal networks connect multiple plant species, facilitating nutrient exchange and communication among trees, a phenomenon sometimes described as the wood wide web. This interconnectedness fosters ecosystem stability and resilience, allowing forests to adapt to environmental changes over time. Additionally, microbial partners can suppress soil-borne diseases, reducing the need for chemical interventions and maintaining the natural balance within these habitats. Such dynamics underscore the role of microbes as ecosystem engineers, quietly shaping the living landscapes that sustain countless forms of life. Recent advances in molecular biology and imaging technologies have deepened our understanding of these symbiotic relationships, revealing complex signaling pathways and genetic exchanges that facilitate communication between plants and microbes. Researchers have discovered that plants release specific chemical signals to attract beneficial microbes, while microbes produce molecules that trigger plant responses, creating a dynamic dialogue essential for establishing and maintaining symbiosis. This knowledge not only enriches our appreciation for the sophistication of microbial ecosystems, but also opens promising avenues for sustainable agriculture. By harnessing natural microbial partnerships, scientists aim to reduce reliance on synthetic fertilizers and pesticides, promoting crop productivity in harmony with environmental health. Such innovations illustrate the potential of microbial symbiosis to inform future strategies for food security and ecosystem conservation. As we continue to explore the diverse strategies microbes employ to thrive alongside plants, it becomes evident that these partnerships are fundamental to life on Earth. The intricate web of interactions between microbial symbionts and plants not only sustains individual organisms, but also orchestrates broader ecological processes vital to planetary health. Moving forward, our attention turns toward other forms of microbial interactions that shape the living world, expanding the horizon of microbial ecology and its profound impact on life's continuity. Beneath the surface of the Earth lies an unseen universe bustling with microscopic life. Soil microbial communities composed of bacteria, fungi, archaea, and other microorganisms play an essential role in maintaining the health and fertility of soils. These tiny organisms act as nature's recyclers, breaking down complex organic matter such as fallen leaves, dead roots, and other plant residues into simpler compounds. This decomposition process releases vital nutrients like nitrogen, phosphorus, and potassium back into the soil, making them accessible to plants and other organisms. Without the diligent work of these microbes, organic materials would accumulate and nutrient cycles would stall, ultimately hindering plant growth and ecosystem productivity. The metabolic activities of soil microbes extend beyond mere decomposition. Many microbes form symbiotic relationships with plant roots, enhancing nutrient uptake and improving soil structure. For example, mycorrhizal fungi extend their hyphae into the soil, increasing the root surface area and helping plants absorb water and minerals more efficiently. Nitrogen-fixing bacteria residing in root nodules of legumes convert atmospheric nitrogen into forms plants can assimilate, enriching the soil naturally without synthetic fertilizers. These interactions foster a resilient soil environment that supports sustainable agriculture by reducing dependency on chemical inputs and promoting long-term soil vitality. Such microbial partnerships exemplify the delicate balance and cooperation that underpin healthy ecosystems. Soil microbes also influence the physical properties of soil, contributing to its structure and porosity. By producing sticky substances called extracellular polymeric substances, microbial communities bind soil particles into aggregates. These aggregates improve soil aeration and water retention, creating an environment conducive to root growth and microbial activity. Furthermore, microbial respiration and biochemical transformations regulate soil pH and redox conditions, which in turn affect nutrient availability and microbial community composition. This dynamic interplay ensures that soils remain fertile and resilient in the face of environmental fluctuations. The extensive networks formed by microbes and their chemical exchanges highlight the soil as a living, breathing entity sustained by countless microscopic interactions. The impact of microbial ecosystems extends beyond local soil health to broader environmental processes. By decomposing organic matter, soil microbes contribute to the global carbon cycle, influencing carbon sequestration and greenhouse gas emissions. Some microbes also participate in the transformation of pollutants, aiding in soil remediation and maintaining ecosystem balance. In agricultural settings, fostering diverse and active microbial communities can enhance crop yields, reduce disease incidence, and support sustainable land management practices. Understanding these microbial dynamics opens pathways for innovative approaches that harmonize human activity with natural biological processes, ultimately sustaining the health of the Earth's soils and the life they nourish. As we observe the profound influence of microbial communities beneath our feet, it becomes clear that these unseen ecosystems are foundational to life's continuity. Their roles in nutrient cycling, symbiotic relationships, soil structure maintenance, and environmental regulation are intricately woven into the fabric of terrestrial ecosystems. Exploring these microbial functions invites us to appreciate the delicate, complex systems that support fertile soils and sustainable agriculture. With this understanding, we turn our attention now to the fascinating ways microbes interact within aquatic environments, expanding the tapestry of microbial influence beyond the soil and into the waters that sustain diverse forms of life. Beneath the vast, shimmering surface of the ocean lies a world teeming with microscopic life forms that silently shape the health and balance of our planet. These marine microbes, invisible to the naked eye, form the foundation of aquatic ecosystems and serve as critical agents in the cycling of nutrients and energy. Among these, phytoplankton, tiny photosynthetic organisms drifting with the currents, play a particularly vital role. Through the process of photosynthesis, phytoplankton converts sunlight into organic matter, producing nearly half of the oxygen we breathe. This unseen army of microorganisms not only sustains marine food webs, but also acts as a global oxygen factory, continuously replenishing the atmosphere. Their ability to fix carbon dioxide into organic carbon links them intimately to the Earth's carbon cycle, influencing patterns of climate regulation that ripple far beyond the ocean's depths. The impact of marine microbes extends well beyond oxygen production. These tiny organisms engage in complex interactions that regulate the flow of carbon through the ocean and atmosphere. When phytoplankton die or are consumed, their organic matter can sink toward the ocean floor, effectively removing carbon from the surface waters and sequestering it in deep-sea sediments, a process known as the biological carbon pump. This natural mechanism helps modulate atmospheric carbon dioxide levels, playing a subtle yet powerful role in mitigating climate change. Additionally, marine bacteria and archaea break down organic matter, recycling nutrients such as nitrogen and phosphorus, which are essential for sustaining life throughout the ocean. These nutrient cycles maintain the productivity of marine ecosystems and support the vast diversity of life they harbor. Moreover, marine microbes contribute to the delicate balance of ocean chemistry and ecosystem health. Certain bacteria form symbiotic relationships with larger marine organisms, such as corals and sponges, aiding in nutrient acquisition and protecting their hosts from disease. Other microbial communities thrive in extreme environments, like hydrothermal vents with a harness chemical energy to support unique ecosystems independent of sunlight. These specialized microbes demonstrate the adaptability and resilience of life, revealing the ocean's capacity to sustain diverse forms of biological activity under varying conditions. Through these myriad roles, microbes underpin the stability and productivity of marine habitats, highlighting their indispensable presence in the aquatic world. The interconnectedness of marine microbial life with global climate processes invites us to reflect on the ocean as a living system, where microscopic organisms act as stewards of planetary health. Changes in microbial community composition or function can have cascading effects on carbon cycling, oxygen production, and nutrient availability, underscoring the sensitivity of these ecosystems to environmental shifts. Understanding the dynamics of marine microbes is therefore essential for predicting how oceans will respond to ongoing climate change and human impacts. As research continues to illuminate these intricate relationships, we gain deeper appreciation for the subtle yet profound ways in which microbial life shapes the world's oceans and, ultimately, the very conditions that make Earth habitable. With this glimpse into the vast influence of microbial ecosystems beneath the waves, we recognize the ocean as a vibrant, living entity, sustained by countless unseen participants. Their delicate interplay with chemical and physical processes forms the foundation for marine biodiversity and global environmental balance. Moving forward, we will explore how these microbial communities interact within freshwater systems, extending our journey into the diverse aquatic realms that nurture life across the planet. Just as microbial communities sustain vast aquatic ecosystems, they are equally vital within the human body, forming what we call the human microbiome. This intricate collection of microorganisms, bacteria, viruses, fungi, and archaea, thrives primarily in the gut, but also inhabits the skin, mouth, and other tissues. These microbes coexist with our cells in a delicate balance, contributing to essential functions that shape our health and well-being. In the gut, they assist in breaking down complex carbohydrates, synthesizing vitamins, and extracting nutrients that our bodies alone could not fully access. This symbiotic relationship exemplifies the profound interdependence between humans and their microscopic partners, revealing a complex ecosystem residing within each of us. The impact of gut microbes extends beyond digestion, reaching into the realm of immunity. By interacting with the cells of the intestinal lining and the immune system, these microorganisms help educate and regulate immune responses, distinguishing between harmful invaders and harmless substances. This modulation supports the body's defense mechanisms while preventing excessive inflammation that can lead to disease. Research increasingly demonstrates that imbalances in microbial communities, known as dysbiosis, may be linked to a variety of conditions including allergies, autoimmune disorders, and infections. Thus, maintaining a healthy and diverse microbiome is crucial for sustaining the body's natural defenses and overall resilience. Beyond physical health, emerging science reveals intriguing connections between the gut microbiome and mental well-being. The gut-brain axis, a complex communication network between the gastrointestinal tract and the central nervous system, allows gut microbes to influence mood, stress responses, and cognitive function. Microbial metabolites can affect neurotransmitter production, immune signaling, and even the permeability of the intestinal barrier, which in turn impacts brain activity. While this field is still unfolding, early findings suggest that nurturing a balanced microbiome might support mental health and offer new approaches to managing conditions such as anxiety and depression. This remarkable link highlights the microbiome's reach far beyond traditional biological boundaries. The human microbiome's role in health exemplifies the broader principle that microbial ecosystems are foundational to life itself. These communities are dynamic, influenced by diet, environment, lifestyle, and genetics, constantly adapting to maintain harmony within their host. As we deepen our understanding of these invisible worlds within us, we gain insight into the interconnectedness of microorganisms and human physiology. This perspective invites a more holistic view of health, where nurturing microbial balance becomes an integral part of personal and medical care. The ongoing exploration of these internal ecosystems promises to unveil new dimensions of biology and medicine, gently reminding us of the profound connections we share with the microbial realm. From the depths of oceanic microbial networks to the intimate ecosystems within our bodies, microbes shape the environments that sustain life in countless ways. As we continue this journey, we will turn our attention to how freshwater microbial communities contribute to the vitality of rivers, lakes, and wetlands, nurturing biodiversity and supporting the intricate web of life on land. These waters, like the oceans and our own bodies, harbor their own unique microbial inhabitants, each playing a vital role in sustaining ecosystems and human societies alike. Beyond the familiar habitats of soil, water, and human bodies, microbial life reveals itself in places once thought inhospitable, even uninhabitable. These extreme environments, ranging from the boiling hot springs of Yellowstone to the crushing pressure of deep-sea hydrothermal vents, harbor astonishing microbial ecosystems that challenge our understanding of life's resilience. Extremophiles, as these organisms are known, thrive under conditions of intense heat, acidity, salinity, or cold, demonstrating an incredible adaptability that expands the known boundaries of biology. In these environments, microbes do not merely survive, they perform essential biochemical processes that sustain complex ecosystems. For instance, in the scalding waters of hot springs, thermophilic bacteria metabolize minerals and gases, creating a foundation for other life forms. These microbial communities often form colorful mats, their vivid hues a testament to the pigments that protect them from harsh ultraviolet light. Such ecosystems reveal how life can persist and flourish in conditions once deemed impossible, inspiring new perspectives on the diversity and tenacity of living organisms. In the abyssal depths of the ocean, where sunlight cannot penetrate and pressures soar to unimaginable levels, microbial life continues its quiet, vital work. Around deep-sea hydrothermal vents, rich plumes of mineral-laden water gush from the Earth's crust, creating pockets of chemical energy that sustain diverse communities of microbes. These chemosynthetic bacteria harness chemical compounds such as hydrogen sulfide to produce organic matter, effectively forming the base of a unique food web independent of solar energy. The microbe's ability to convert inorganic molecules into life-sustaining nutrients illustrates an alternative pathway for energy flow on our planet. This remarkable adaptation not only supports a variety of vent-dwelling creatures, such as tube worms and crustaceans, but also reshapes our conception of where life can originate and persist. Studying these deep-sea microbial ecosystems offers profound insights into Earth's biogeochemical cycles and the potential for life in similar extreme environments beyond our planet. On the opposite extreme of temperature, microbial ecosystems also flourish within the frozen landscapes of Antarctica. Beneath thick layers of ice, in subglacial lakes and briny pockets, psychrophilic microorganisms have adapted to survive in perpetual cold, scarce nutrients, and low light. These cold-loving microbes utilize specialized enzymes and cellular structures to maintain metabolic functions despite the harsh freeze. Their presence within icicles and sediment layers reveals a hidden biosphere that participates in nutrient recycling and influences global climate processes. Additionally, Antarctic microbial life illustrates the remarkable plasticity of life's chemical and physical requirements, offering clues about how life might exist in icy worlds elsewhere in the solar system. The study of such ecosystems enriches our appreciation of microbial diversity and resilience, emphasizing that even the coldest, darkest habitats on Earth are alive with microscopic activity. Collectively, these microbial ecosystems in extreme environments underscore the incredible versatility of life and its capacity to adapt to seemingly impossible conditions. Extremophiles not only expand the definition of life's limits, but also provide invaluable knowledge about evolutionary processes, biochemistry, and potential applications in biotechnology. Their unique metabolic pathways and survival strategies inspire innovations in medicine, environmental remediation, and industrial processes. Moreover, the existence of life in such extreme niches invites us to contemplate the possibilities of extraterrestrial life, encouraging exploration of planets and moons where similar conditions might prevail. As we deepen our understanding of these resilient microbial communities, we begin to see life as a continuum capable of thriving across an astounding range of environments. As we continue to explore the myriad ways microbes influence their surroundings, we now turn our attention to the vital role of microbial communities within freshwater systems. Rivers, lakes, and wetlands each host their own distinct assemblies of microscopic life that contribute profoundly to ecosystem health and biodiversity. These freshwater microbes mediate nutrient cycles, support food webs, and help maintain water quality, linking the microbial world intimately with the landscapes and organisms that depend on them. Understanding these dynamic communities offers a glimpse into the delicate balance sustaining life above and below the water's surface. Beneath the surface of every environment, from the richest soils to the vast depths of the oceans, microbial communities quietly orchestrate the essential cycles that sustain life on Earth. These invisible organisms are the unseen engineers of biogeochemical processes, driving the continuous movement and transformation of elements such as carbon, nitrogen, sulfur, and phosphorus. Through their metabolic activities, microbes regulate the availability of these critical nutrients, ensuring that ecosystems remain balanced and productive. For instance, in the carbon cycle, microbes decompose organic matter, releasing carbon dioxide back into the atmosphere or transforming it into forms that support plant growth. This delicate interplay maintains the equilibrium between carbon storage and release, influencing global climate patterns. Nitrogen, a fundamental building block of proteins and nucleic acids, exists abundantly in the atmosphere, but in a form unusable by most living organisms. Here, specialized bacteria and archaea perform the remarkable feat of nitrogen fixation, converting inert atmospheric nitrogen into ammonia through enzymatic processes. This ammonia is then further processed by other microbes in nitrification and denitrification steps, cycling nitrogen through the soil and water, making it accessible to plants and eventually to animals. Without these microbial transformations, the nitrogen cycle would stall, severely limiting life's capacity to flourish. These processes are subtle yet vital, linking the biosphere and atmosphere in a continuous exchange that sustains all terrestrial and aquatic life. Beyond carbon and nitrogen, sulfur and phosphorus cycles are equally essential, each intricately managed by a variety of microbial agents. Sulfur-transforming bacteria catalyze the conversion of sulfur compounds through oxidation and reduction reactions, impacting everything from soil fertility to the chemistry of natural waters. These transformations influence the availability of sulfur nutrients and affect the formation of minerals and atmospheric gases. Similarly, phosphorus, often a limiting nutrient in ecosystems, is mobilized and recycled by microbial activity. Microbes break down organic phosphorus compounds, releasing phosphate ions that plants and other organisms rely upon for growth and energy transfer. The interconnectedness of these cycles, mediated by microbial life, highlights the profound influence of microorganisms on Earth's environmental balance. The complexity and resilience of microbial ecosystems lie in their adaptability and diversity, allowing biogeochemical cycles to function under a wide range of conditions. Whether in oxygen-rich surface waters or anoxic sediments, microbes adjust their metabolic pathways to maintain nutrient flows. This adaptability also enables ecosystems to recover from disturbances such as pollution or climate fluctuations by restoring nutrient availability and supporting the food webs that depend on them. As we deepen our understanding of these microbial processes, we begin to appreciate the subtle, yet powerful ways in which microbes sustain life's interconnected web, shaping environments both large and small. Appreciating the role of microbial communities in driving biogeochemical cycles invites us to consider the broader implications for ecosystem management and conservation. By recognizing how microbial processes underpin nutrient availability and environmental health, researchers and policymakers can better address challenges such as soil degradation, water quality decline, and climate change. The study of these tiny yet mighty organisms continues to reveal new insights into the delicate balances that keep our planet thriving. As we reflect on this microbial stewardship of elemental cycles, we prepare to explore their specific contributions within the freshwater environments we first encountered, where these processes unfold in intricate and remarkable ways. Microbial communities, though invisible to the naked eye, exert profound influence over the Earth's climate system. These microscopic organisms participate actively in the production and consumption of greenhouse gases such as carbon dioxide, methane, and nitrous oxide. In wetlands, for example, methanogenic archaea generate methane under anaerobic conditions, contributing to one of the most potent greenhouse gases in our atmosphere. Conversely, other groups of microbes consume methane, mitigating its release into the environment. This delicate balance between microbial production and consumption of greenhouse gases shapes atmospheric composition and thus climate dynamics. Understanding these microbial processes provides critical insights into natural feedback mechanisms that either amplify or dampen climate change. As the planet warms and ecosystems shift, the activity and distribution of these microbial populations evolve, influencing the magnitude and direction of greenhouse gas fluxes. Beyond their role in greenhouse gas emissions, microbes serve as vital agents of carbon sequestration, capturing atmospheric carbon and transforming it into stable organic matter. In soils and aquatic sediments, microbial communities decompose organic materials, some of which become incorporated into long-term carbon reservoirs. The complex interactions between microbes and their environments determine how much carbon is stored versus released, affecting the global carbon cycle. Certain bacteria and fungi form symbiotic relationships with plants, enhancing root growth and soil structure, which further promotes carbon storage. These microbial-mediated processes not only influence carbon availability for other organisms, but also help regulate atmospheric carbon dioxide levels. As research advances, it becomes clear that microbes contribute significantly to the Earth's natural capacity to mitigate climate change through carbon sequestration, highlighting their indispensable role in ecosystem resilience. The intricate relationship between microbial activity and climate extends to feedback loops that can either stabilize or destabilize environmental conditions. For instance, thawing permafrost releases previously frozen organic matter, which microbes then decompose, emitting greenhouse gases and accelerating warming. Conversely, some microbial pathways can enhance soil fertility and plant growth, increasing photosynthetic carbon uptake and offsetting emissions. These dynamic interactions underscore the complexity of predicting climate trajectories, as microbial responses to environmental changes are not yet fully understood or integrated into many climate models. Scientists now recognize the necessity of incorporating microbial ecology into predictive frameworks to improve the accuracy of climate forecasts. By doing so, they aim to better anticipate the potential consequences of microbial feedbacks on global warming and inform more effective mitigation strategies. In aquatic ecosystems, microbes likewise influence climate processes through their control of carbon and nutrient cycling. Phytoplankton, microscopic photosynthetic organisms, fix vast amounts of carbon dioxide, forming the foundation of marine food webs and driving the biological carbon pump. When these organisms die, some of their biomass sinks to the ocean floor, sequestering carbon for centuries or longer. Additionally, microbial decomposition in water bodies releases greenhouse gases, linking aquatic systems closely to atmospheric chemistry. Changes in temperature, nutrient availability, and oxygen levels affect microbial community composition and function, thereby altering their climatic impact. Through such mechanisms, microbial ecosystems in freshwater and marine environments represent crucial components of the Earth's climate puzzle, where subtle shifts can cascade into broader environmental consequences. As we deepen our understanding of microbial contributions to greenhouse gas dynamics and carbon storage, it becomes evident that these organisms are integral to the feedbacks governing climate change. Their dual role, both as sources and sinks of greenhouse gases, presents both challenges and opportunities for managing our planet's future. Incorporating microbial processes into climate models enhances our ability to predict environmental outcomes and develop informed conservation strategies. By appreciating the multifaceted roles microbes play in regulating Earth's atmosphere, we move closer to a holistic perspective on ecosystem health and climate resilience. This awareness invites us to explore further how microbial ecosystems function within specific habitats, such as the freshwater environments, where their impact is both profound and complex. As we explore the remarkable versatility of microbial life, it becomes clear that these microscopic organisms are not only fundamental to natural ecosystems, but also hold immense promise for advancing human technology. Recent research has illuminated how microbes can be harnessed for biotechnological applications, transforming fields such as bioenergy, environmental remediation, and synthetic biology. For instance, certain bacteria and algae possess the ability to convert organic materials into biofuels, offering renewable alternatives to fossil fuels that could reduce our carbon footprint. These biofuels, derived from microbial metabolism, are produced through processes that are often more sustainable and environmentally friendly than conventional energy sources. The potential for scaling such microbial biofuel production continues to inspire scientists and engineers alike, as it aligns with global efforts to transition toward cleaner energy. Beyond energy, microbes also play a critical role in waste treatment and environmental cleanup. specialized microbial communities can degrade pollutants, breaking down complex chemical compounds into harmless substances. This natural biodegradation process forms the foundation of many wastewater treatment systems, where microbes remove organic waste and neutralize harmful chemicals before water is released back into the environment. Emerging research seeks to optimize these microbial consortia, engineering them to target an even wider array of contaminants, including heavy metals and synthetic compounds that resist conventional treatment. By tapping into the metabolic diversity of microbes, biotechnology offers practical solutions for mitigating pollution and preserving water quality. Harnessing these microbial capabilities not only supports ecosystem health, but also contributes to public health and sustainable urban development. In the realm of synthetic biology, microbes serve as living factories, engineered to produce valuable compounds ranging from pharmaceuticals to biodegradable plastics. Advances in genetic engineering have enabled scientists to redesign microbial genomes, programming them to perform specific functions with precision and efficiency. This innovative approach leverages the rapid growth rates and adaptability of microbes, allowing for scalable production processes that are both cost-effective and environmentally conscious. Synthetic biology also opens doors to creating entirely new biological systems, expanding the possibilities for innovation in medicine, agriculture, and materials science. As we develop these microbial platforms, ethical considerations and safety measures remain paramount, ensuring that these powerful tools are used responsibly for the benefit of society. The intersection of microbiology and engineering thus heralds a new era of technological progress, rooted in the intricate biology of microorganisms. Looking ahead, the integration of microbial ecosystems into emerging technologies underscores a broader trend toward biomimicry, drawing inspiration from natural processes to solve complex human challenges. Microbes, with their extraordinary metabolic diversity and ecological roles, provide blueprints for sustainable innovation that harmonizes with Earth's systems. Research continues to uncover novel microbial species and pathways that could revolutionize industries and contribute to circular economies. Such discoveries emphasize the importance of preserving microbial diversity, as each organism may hold unique potential for future applications. As we venture deeper into this microscopic frontier, interdisciplinary collaboration will be essential to fully realize the benefits of microbial biotechnology while maintaining ecological balance. This ongoing journey invites us to reconsider our relationship with the microbial world, recognizing it as a vital partner in shaping a sustainable future. As these scientific endeavors unfold, the profound implications of microbial biotechnology extend beyond technical achievements, touching upon societal and environmental dimensions. The ability to engineer microbial systems for targeted purposes challenges us to think critically about resource use, ethical frameworks, and equitable access to technology. It also offers hope for addressing some of the most pressing issues of our time, from climate change mitigation to global health. By fostering a deeper understanding of microbial ecosystems and their capabilities, we prepare to harness their full potential in ways that respect the interconnectedness of life on Earth. This evolving perspective naturally leads us to consider specific habitats where microbes operate, such as the complex freshwater environments teeming with microbial diversity, and how these settings influence both natural processes and technological innovation. Microbial ecosystems weave an intricate tapestry of interactions within and around living organisms, profoundly influencing health and disease. Among these microscopic communities are pathogenic microbes, agents capable of causing illness by invading hosts and disrupting physiological harmony. Yet the story of microbes and disease is not simply one of harm. Instead, it reveals a delicate balance where microbial inhabitants coexist, compete, and collaborate, shaping the environment of the host in ways that can both prevent and promote disease. These ecosystems are remarkably complex, with countless species interacting in dynamic networks that respond to changes in the host's internal and external conditions. Understanding this complexity is essential, as it guides us in discerning how disruptions to microbial balance, often termed dysbiosis, can lead to vulnerability to infection, chronic inflammation, or other health challenges. Within the human body, microbial ecosystems inhabit diverse niches, from the skin surface to the depths of the gut, each contributing uniquely to health maintenance. Beneficial microbes engage in vital functions such as aiding digestion, synthesizing essential vitamins, and training the immune system to distinguish friend from foe. When these microbial communities remain balanced, they act as a natural defense, outcompeting or inhibiting the growth of harmful pathogens through mechanisms like nutrient competition and the production of antimicrobial substances. However, when this equilibrium is disturbed by factors such as antibiotic overuse, dietary changes, or illness, opportunistic pathogens may gain dominance, disrupting host health and triggering disease states. This interplay underscores the importance of viewing disease not merely as the presence of harmful microbes, but as a breakdown in the harmonious relationships within microbial ecosystems. The complexity of microbial ecosystems extends beyond human hosts, influencing disease processes across diverse environments and species. In natural habitats, microbial communities regulate the spread and persistence of pathogens among plants, animals, and even in aquatic systems. For example, soil microbes can suppress plant diseases by creating unfavorable conditions for pathogens, thereby supporting agricultural productivity and ecosystem resilience. Similarly, in aquatic environments, microbial interactions affect the emergence of waterborne diseases by mediating nutrient cycles and influencing host susceptibility. These interconnected systems illustrate how microbial balance serves as a critical factor in disease ecology, where shifts in microbial composition can reverberate through food webs and impact ecosystem health. Such insights encourage a holistic approach to studying disease, one that appreciates microbial ecosystems as integral to understanding and managing health risks in both clinical and environmental contexts. Advances in microbial biotechnology and metagenomic analysis have illuminated the vast diversity and functional potential of microbial ecosystems, revealing how subtle changes in microbial populations can have profound implications for health. By mapping these communities and their interactions, researchers are uncovering new strategies for disease prevention and treatment that aim to restore or maintain microbial balance rather than merely eliminating pathogens. Probiotic therapies, microbiome transplants, and precision antibiotics are examples of approaches emerging from this nuanced understanding. These interventions reflect a shift from viewing microbes solely as enemies to recognizing their essential role in sustaining health. Yet such efforts also remind us of the ecosystem's fragility and the need for careful stewardship to avoid unintended consequences. The ongoing exploration of microbial ecosystems thus continues to reshape our relationship with the microscopic world and informs future directions in medicine and environmental management. As we deepen our exploration of microbial ecosystems and their multifaceted roles in health and disease, it becomes clear that these communities embody a profound complexity that challenges simplistic notions of good and bad microbes. This perspective invites us to appreciate the delicate balance maintained within microbial habitats and the ways in which ecological disruptions can propagate disease. It also highlights the potential for innovative solutions rooted in the restoration and preservation of microbial harmony. Looking ahead, the study of microbial ecosystems offers promising avenues for understanding how life itself is shaped by invisible networks, guiding us toward more integrated approaches to health and environmental stewardship. With this understanding, we turn our attention to specific microbial habitats, such as the diverse and dynamic freshwater ecosystems, where microbial life not only thrives, but also drives essential biogeochemical processes. Within the intricate webs of microbial ecosystems, communication plays a fundamental role in shaping community behavior and resilience. Unlike multicellular organisms that rely on nervous or hormonal systems to coordinate actions, microbes employ chemical signals to exchange information across their populations. This phenomenon, known as quorum sensing, allows individual cells to sense their population density and collectively regulate gene expression in response to changing environmental conditions. Through the release and detection of small signaling molecules, microbes can synchronize activities that would be ineffective if performed by solitary cells. Such coordination enables them to adapt dynamically, whether by modulating nutrient uptake, producing antimicrobial compounds, or adjusting motility. The subtle language of microbial communication reveals a remarkable level of organization, demonstrating that these microscopic entities are far from simple, isolated units, but are instead members of cooperative networks that enhance their survival and ecological success. One of the most striking outcomes of microbial communication is the formation of biofilms, structured communities of microbes adhering to surfaces and encased within a self-produced matrix of extracellular polymeric substances. Through quorum sensing, microbes collectively decide when to initiate biofilm development, a process that offers protection against environmental stresses, antibiotics, and immune responses. In natural and artificial environments alike, biofilms influence nutrient cycling, water quality, and even the progression of infectious diseases. These microbial cities demonstrate how communication fosters complexity, allowing cells to transition from free-floating individuals to organized communities with specialized functions. The matrix not only anchors the population, but also facilitates the exchange of nutrients and genetic material, further enhancing the adaptability of the community. As we begin to appreciate the sophistication of these microbial interactions, it becomes evident that signaling molecules are not merely chemical messengers, but pivotal agents in the orchestration of life at the microscopic scale. Beyond biofilm formation, microbial signaling mediates diverse processes essential to ecosystem functioning. In freshwater habitats, for example, communication among bacteria and algae can regulate the timing of nutrient uptake and photosynthesis, optimizing resource use in fluctuating conditions. Similarly, soil microbes use signaling pathways to coordinate the degradation of organic matter, influencing carbon cycling and soil fertility. These interactions extend to complex symbiotic relationships where microbial partners communicate with plant roots or animal hosts to establish mutualistic associations. Through such dialogues, microbes can modulate host immune responses, promote growth, or protect against pathogens. This intricate exchange highlights how microbial communication is a cornerstone of ecological balance, enabling cooperation that supports the health of entire ecosystems. Observing these processes invites us to consider the invisible dialogues that sustain life, reminding us of the interconnectedness that exists far beneath the surface of what is immediately visible. Advances in molecular biology and imaging technologies have illuminated the pathways and molecules involved in microbial signaling, revealing a diversity of chemical languages used by different species. From acyl homoserine lactones in gram-negative bacteria to oligopeptides in gram-positive bacteria, these signals differ in structure but converge in function to inform, coordinate, and regulate. The specificity and sensitivity of these systems allow microbes to distinguish friend from foe, modulate competitive behaviors, and even manipulate host physiology. Understanding these signaling networks opens new possibilities in medicine and environmental management, such as disrupting harmful biofilms or enhancing beneficial microbial functions. As research continues to decode the complexity of microbial communication, it broadens our appreciation of microbes not as isolated organisms, but as participants in elaborate social networks that influence health, environment, and evolution. This growing knowledge guides us toward innovative strategies that harness microbial cooperation rather than confrontation. As we continue to explore the nuanced roles of microbial communication within ecosystems, it becomes clear that these signaling processes are vital to the maintenance of microbial diversity and function. They enable communities to respond collectively to environmental stresses, to synchronize metabolic activities, and to form structured assemblies that shape habitat conditions. Such dynamics are especially evident in freshwater ecosystems, where microbial signaling governs interactions that drive nutrient cycling, organic matter decomposition, and the overall productivity of the water column. The delicate balance maintained through these chemical conversations underscores the importance of preserving microbial habitats, as disruptions can cascade through ecosystems with profound consequences. With a deeper understanding of microbial communication, we move closer to appreciating the invisible forces that sustain life and the potential for leveraging these processes in environmental stewardship. Turning now to the specific habitats where these microbial networks flourish, we consider the remarkable diversity and activity within freshwater microbial communities. As we delve deeper into the world of microbial ecosystems, it is impossible to overlook the profound ways in which human activities influence these microscopic communities. Pollution, in its many forms, introduces a variety of chemical substances into natural environments that can alter microbial diversity and function. Heavy metals, pesticides, and nutrient runoff from agricultural landscapes disturb the delicate chemical balance that microbes rely upon for communication and cooperation. These contaminants may inhibit beneficial microbial processes, such as nitrogen fixation or organic matter decomposition, while encouraging the growth of opportunistic species that can upset established ecological equilibriums. The consequences of these shifts extend beyond microbial realms, affecting plant health, water quality, and the broader food web. Recognizing that microbial communities are sensitive indicators of environmental change prompts a deeper awareness of the interconnectedness between human actions and microbial well-being. Understanding this link is essential as we consider the stewardship of ecosystems that are often unseen, yet critical to planetary health. Antibiotics, vital for combating infectious diseases, represent another significant human influence on microbial ecosystems. When introduced into the environment through medical waste or agricultural use, these compounds can disrupt natural microbial populations by selectively inhibiting susceptible bacteria and allowing resistant strains to thrive. This selective pressure not only diminishes microbial diversity, but also contributes to the emergence and spread of antibiotic resistance genes, which pose a serious challenge to both environmental and human health. In aquatic and soil environments, antibiotic residues affect microbial communication pathways, potentially impairing processes like quorum sensing that regulate community behavior and resilience. The ripple effect of such disruptions can weaken ecosystem functions, including pollutant degradation and nutrient cycling. As we become more aware of these impacts, it becomes clear that managing antibiotic use and disposal is an important step in safeguarding microbial ecosystems and the services they provide. Such awareness invites a broader conversation about the responsible integration of human technologies with natural microbial processes. Habitat destruction, driven by urban expansion, deforestation, and land use changes, further threatens the integrity of microbial ecosystems. The physical alteration or loss of habitats interrupts the spatial structure and connectivity that microbial communities depend upon for exchange and cooperation. Soil compaction, changes in moisture regimes, and the reduction of organic matter input all contribute to shifts in microbial composition and activity. In freshwater systems, sediment disruption and altered flow patterns impact microbial biofilms and nutrient dynamics, undermining the stability of these finely tuned environments. The loss of microbial habitat diversity reduces the overall resilience of ecosystems to environmental stresses, making them more vulnerable to invasive species and climate variability. This cascade of effects highlights the importance of conserving not only visible flora and fauna, but also the microscopic organisms that underpin ecosystem health. Fostering strategies that protect and restore microbial habitats is therefore essential in maintaining the vitality of natural landscapes. The interplay between human influence and microbial ecosystems gently reminds us of the fragility and complexity of life's smallest architects. As pollutants seep into soils and waters, as antibiotics permeate environments, and as habitats are fragmented or lost, microbial communities face unprecedented challenges. Yet these communities possess remarkable adaptability, often finding ways to adjust signaling mechanisms, metabolic pathways, and community structures in response to change. The resilience observed in microbial ecosystems offers hope, but it also emphasizes the need for thoughtful conservation efforts that minimize disturbance and support natural recovery processes. By integrating microbial perspectives into environmental management, we can develop more holistic approaches that honor the subtle yet essential roles microbes play. This perspective encourages a shift from viewing microbes merely as isolated entities toward appreciating them as dynamic participants in the larger web of life. Looking ahead, the challenge lies in deepening our understanding of how specific human actions influence microbial networks across diverse ecosystems. Advances in molecular biology and environmental monitoring provide tools to observe these changes with increasing precision, revealing complex feedback loops and previously hidden interactions. Through such knowledge, we can better predict the consequences of pollution, antibiotic presence, and habitat alteration on microbial functions critical to ecosystem sustainability. This growing awareness not only informs conservation policies, but also inspires innovative solutions that work in harmony with microbial processes. As we continue to unravel these intricate relationships, we prepare to foster environments where microbial communities can thrive alongside human development. Moving forward, it is valuable to explore particular habitats where these delicate balances are tested and conserved in equal measure. In the delicate dance of life, microbial ecosystems serve as unseen architects, shaping the health of soils, waters, and even the air we breathe. When these intricate communities face disruption, whether through pollution, chemical spills, or the overuse of antibiotics, the delicate balance that sustains their vital functions becomes compromised. Yet, within this challenge lies a remarkable opportunity: the ability to restore these ecosystems through intentional, science-driven efforts. Bioremediation emerges as a powerful tool in this endeavor. By harnessing specific microorganisms known for their capacity to degrade harmful substances, scientists can guide the natural breakdown of pollutants, transforming contaminated environments back toward equilibrium. These microbial agents work invisibly but effectively, breaking down oil residues, heavy metals, and synthetic chemicals into less harmful forms. Their activity exemplifies the profound resilience embedded within microbial networks and offers hope that even severely damaged habitats can recover their functional integrity. As we deepen our understanding of microbial capabilities, the scope of bioremediation expands, inviting new strategies tailored to diverse environmental challenges. Alongside bioremediation, probiotics represent another avenue through which microbial balance may be gently restored, particularly when considering the health of living organisms and their surrounding environments. In both agricultural and medical contexts, introducing beneficial microbes can support weakened or disrupted microbial communities, promoting stability and resilience. These probiotics, carefully selected for their positive interactions, act as guardians of microbial diversity, outcompeting harmful pathogens and enhancing nutrient cycling. In soils, for example, probiotic treatments can stimulate plant growth by improving nutrient availability and suppressing disease-causing organisms, thereby contributing to sustainable farming practices that minimize chemical inputs. Similarly, in aquatic systems, probiotic approaches help maintain water quality by balancing microbial populations that regulate organic matter decomposition. This subtle reintroduction of microbial allies underscores the interconnectedness of microbial ecosystems and the organisms they support, revealing a path toward harmony rather than dominance. The gentle restoration of these communities reflects a growing recognition that microbial health is integral to broader environmental and human well-being. The promise of restoring microbial balance through bioremediation and probiotics invites us to reimagine our relationship with the microscopic world. It encourages a shift from viewing microbes solely as agents of disease or pollution toward acknowledging their essential roles in ecosystem function and recovery. Advanced molecular techniques allow us to identify specific microbial strains capable of addressing particular environmental challenges, fostering targeted interventions that work synergistically with natural processes. This approach not only enhances the efficiency of restoration efforts, but also minimizes unintended consequences, preserving the richness of microbial diversity that underpins ecosystem resilience. Moreover, the integration of microbial management into environmental policy and land use planning reflects a holistic perspective, one that values the invisible yet indispensable contributions of microbial life. As these innovative strategies continue to evolve, they offer a beacon of hope for healing landscapes altered by human activity, guiding us toward a future where microbial ecosystems regain their balance and vitality. Unlocking the mysteries of microbial ecosystems has long been a challenge due to the sheer invisibility and complexity of these communities. Traditional culture-based methods, while foundational, capture only a fraction of microbial diversity, leaving countless species and interactions unexplored. In recent decades, however, revolutionary advancements in metagenomics have transformed our ability to peer into these hidden worlds. By sequencing the collective genetic material from environmental samples, metagenomics bypasses the need to culture individual organisms, revealing a rich tapestry of microbial life in soils, oceans, and even within living hosts. This approach not only catalogues microbial diversity with unprecedented depth, but also allows scientists to infer functional capacities, metabolic pathways, and ecological roles of these organisms. Through metagenomic analyses, we are beginning to appreciate the vast interconnected networks that sustain nutrient cycling, energy flow, and environmental stability, processes critical to life on Earth. Alongside metagenomics, advances in microscopy have provided complementary insights, offering visual windows into microbial form and behavior. High-resolution imaging techniques such as confocal laser scanning microscopy and cryo-electron microscopy enable us to observe microbes in their native habitats with remarkable clarity. These tools reveal intricate cellular structures, biofilm formations, and dynamic interactions among microbial cells and their environment. Fluorescent tagging methods further allow researchers to track specific species or metabolic activities in situ, deepening our understanding of microbial ecology. Together, these imaging technologies illuminate the spatial organization and temporal dynamics of microbial communities, bridging the gap between genetic potential and ecological function. Such visualizations remind us that microbial ecosystems are not static, but vibrant, ever-changing systems that respond sensitively to environmental cues. The integration of metagenomics and advanced microscopy represents a powerful fusion of molecular and visual science, enabling a holistic perspective on microbial ecosystems. Through this synergy, researchers can link genetic information with physical context, deciphering how microbes interact with one another and with their surroundings. For instance, these tools have uncovered cooperative networks within microbiomes that enhance nutrient availability or confer resilience against stress. They have also identified keystone species whose presence disproportionately influences ecosystem stability and function. This knowledge is essential for designing effective interventions to restore or manipulate microbial communities in agriculture, medicine, and environmental management. As technology continues to evolve, so too does our capacity to explore the unseen microbial world with precision and care, unveiling layers of complexity that were once beyond our grasp. Moreover, the accessibility of these cutting-edge tools has democratized microbial research, fostering interdisciplinary collaborations and citizen science initiatives. Portable sequencing devices and user-friendly imaging platforms empower researchers across the globe to contribute to the growing repository of microbial knowledge. This collective effort enriches our understanding of microbial diversity across diverse ecosystems and under varying conditions. It also accelerates the discovery of novel species and bioactive compounds with potential applications in biotechnology and medicine. By embracing technological innovation, we are not only expanding the frontiers of microbial ecology, but also nurturing a deeper appreciation for the intricate web of life that thrives beyond the limits of human vision. The unfolding revelations from these studies invite us to reconsider the fundamental connections between microbes and planetary health. As we delve deeper into the microbial realm through these sophisticated methods, the boundaries between disciplines blur and new questions emerge. How do microbial communities adapt to rapid environmental change? What roles do viruses and rare microbial species play in ecosystem resilience? Can we harness microbial capabilities to mitigate climate change or enhance sustainable agriculture? The continuous refinement of metagenomic sequencing and microscopy offers promising pathways to address these inquiries. These tools serve as keys to unlock the profound influence of microbes on global biogeochemical cycles and human well-being. The journey into the microbial unknown, illuminated by technology, guides us toward a future where invisible life forms are understood not only as subjects of study, but as vital partners in sustaining the living world. Microbial ecosystems are among the most ancient and fundamental components of life on Earth. Long before the rise of complex multicellular organisms, microscopic life forms inhabited every conceivable environment, from the depths of the oceans to volcanic hot springs. These tiny architects of life played a pivotal role in shaping the planet's early atmosphere and geochemical cycles, setting the stage for the biological diversity we observe today. In considering the origin of life itself, microbes provide a living bridge to the earliest forms of life, offering clues about how simple molecules may have assembled into self-replicating entities. The study of modern microbial communities in extreme environments, such as hydrothermal vents and acidic springs, allows scientists to infer possible scenarios where life first emerged. These ecosystems exhibit remarkable metabolic versatility, harnessing chemical energy from inorganic sources in the absence of sunlight, a trait that may echo the conditions of primordial Earth. Through these insights, microbes are not only subjects of biological inquiry, but also windows into the processes that gave rise to life's very beginnings. The significance of microbial ecosystems extends beyond their ancient origins to their ongoing influence on Earth's biosphere. Microbes participate in essential biogeochemical cycles, including those of carbon, nitrogen, and sulfur, regulating the flow of elements that sustain life. Their metabolic activities have transformed Earth's atmosphere over geological timescales, notably through the production of oxygen via photosynthetic cyanobacteria. This oxygenation event, often called the Great Oxidation Event, dramatically altered the trajectory of biological evolution, enabling the rise of aerobic organisms and complex life forms. In this way, microbial ecosystems have functioned as both creators and maintainers of habitable conditions on our planet. By examining the interactions within these communities, scientists gain a richer understanding of how life's complexity evolved from simple beginnings. This perspective underscores the fundamental role microbes hold in Earth's biological history, linking them directly to hypotheses about life's inception and persistence. The exploration of microbial life also intersects with the search for life beyond Earth. Understanding how microbial ecosystems adapt and thrive in extreme terrestrial environments informs astrobiology, guiding the search for potential biosignatures on other planets and moons. The resilience and metabolic diversity displayed by these microscopic organisms suggest that life could exist in conditions once thought inhospitable. By studying microbial ecosystems that resemble early Earth or extraterrestrial environments, such as subsurface ice or acidic lakes, researchers develop models for how life might originate and sustain itself elsewhere in the cosmos. This scientific journey links the microscopic world intimately with profound questions about life's universality and origins. Thus, microbial ecosystems serve as both a reference point for Earth's past and a beacon for understanding life's potential beyond our home planet. Advances in molecular biology and genomics have deepened our appreciation for the complexity of microbial communities and their evolutionary significance. Metagenomic analyses reveal vast networks of genetic exchange among microbes, illustrating how horizontal gene transfer accelerates adaptation and diversification. Such mechanisms may have been crucial during Earth's early epochs, facilitating the emergence of novel metabolic pathways and ecological strategies. These genetic interactions within microbial ecosystems provide a dynamic framework for understanding how life's earliest forms could have evolved from simple replicators into the diverse array of organisms present today. The continuous study of these processes enriches our comprehension of the fundamental principles governing life's origin and resilience. In this way, microbes not only illuminate the past, but also shape our understanding of biology's ever-evolving narrative. As the journey into microbial ecosystems unfolds, it becomes clear that these microscopic communities are integral to the story of life itself. Their presence, diversity, and metabolic ingenuity highlight the profound connections linking biology, geology, and chemistry across deep time. The ongoing exploration of these systems invites us to reconsider life's complexity from a microbial perspective, appreciating the subtle but powerful roles these organisms have played in Earth's history. By embracing this viewpoint, scientific inquiry moves toward a more holistic understanding of life's origins, poised to uncover further revelations about the intricate web of existence. This exploration sets the stage for a deeper investigation into the interactions between microbes and their environments, revealing the delicate balance that sustains ecosystems at every scale. Microbial ecosystems may seem distant from our everyday experiences, yet their influence reaches into the very fabric of our lives. These tiny communities, composed of bacteria, archaea, fungi, and other microorganisms, perform essential tasks that sustain the health of soil and crops. Through processes like nitrogen fixation, microbes convert atmospheric nitrogen into forms usable by plants, enabling the growth of the food that nourishes us. Without this crucial function, agriculture would struggle to meet the needs of a growing population. Beyond the fields, microbes actively contribute to the decomposition of organic matter, recycling nutrients that maintain fertile landscapes. Their activity supports the delicate balance of ecosystems, ensuring that plants and animals alike have the resources necessary to thrive. This intricate cooperation between microbes and the environment underscores how intimately microbial health is tied to the security of our food systems and the stability of natural habitats. The impact of microbial ecosystems extends far beyond agriculture, reaching into the realm of human medicine. Our bodies are home to trillions of microorganisms, collectively known as the microbiome. which play a vital role in digestion, immune function, and even mental well-being. These microbial communities form protective barriers against harmful pathogens and assist in breaking down complex foods, enhancing nutrient absorption. Scientific advances have begun to reveal how disruptions in our microbiome can contribute to a range of health issues, from allergies to chronic diseases. The study of these microbial relationships is opening new avenues for treatments that harness the power of beneficial microbes, offering hope for personalized medicine and improved healthcare outcomes. In this way, the unseen world within us reflects the larger microbial ecosystems that shape the environment outside our bodies. On a planetary scale, microbial ecosystems are indispensable actors in the regulation of Earth's climate. Microbes participate in carbon cycling by breaking down organic matter and releasing or sequestering greenhouse gases such as carbon dioxide and methane. Wetlands, forests, and oceans host diverse microbial communities that influence the balance of these gases in the atmosphere, thereby affecting global temperatures and weather patterns. Changes to microbial populations, driven by human activity or environmental shifts, can alter these processes with far-reaching consequences. Understanding the roles microbes play in climate stability is essential for developing sustainable strategies to mitigate climate change and protect vulnerable ecosystems. This knowledge highlights the interconnectedness of all life forms and the subtle yet powerful ways in which microbes help maintain the equilibrium of our planet's systems. Considering these connections, it becomes clear that microbial ecosystems are not isolated curiosities, but foundational to the health of agriculture, medicine, and climate. Their complex interactions sustain the resources we depend upon, from the food on our tables to the air we breathe. By appreciating the importance of microbial health, we are invited to think more deeply about how human actions influence these communities and the broader environment. This perspective encourages mindful stewardship and inspires innovations that align with the natural processes shaped by microbes over billions of years. As we deepen our understanding of these living networks, the potential to enhance human well-being and environmental resilience grows ever more promising. The significance of microbial ecosystems lies not only in their vast numbers or ancient origins, but in their ongoing contributions to the vitality of the planet and ourselves. Each microscopic organism plays a part in a larger symphony that supports life's complexity and diversity. Recognizing this invites a shift in how we perceive our relationship to the natural world, fostering respect for the unseen forces that sustain us. This awareness opens a gateway to exploring the delicate interplay between microbes and their environments, setting the stage for further investigation into the mechanisms that balance ecosystems and influence global health. From this vantage point, the journey into microbial ecosystems continues, revealing ever more intricate connections that shape the future of life on Earth. As we draw our reflections on microbial ecosystems to a close, it becomes clear that these tiny architects of life invite us into a deeper understanding of existence itself. Far beyond mere curiosity, embracing the microbial world means acknowledging the intimate ties that bind all living things in a shared web of survival. These microorganisms, often invisible to the naked eye, orchestrate processes that regulate climate, We cycle nutrients and sustain the health of every ecosystem on the planet. Their resilience and adaptability offer lessons in balance and renewal, reminding us that life's persistence depends on cooperation at every scale. In this light, our stewardship of microbial habitats is not simply an environmental responsibility, but a profound act of respect for the intricate foundations of life. By opening our hearts and minds to the silent symphony of microbes, we nurture a sense of wonder and gratitude that can inspire sustainable choices and a harmonious coexistence with the natural world. The impact of microbial ecosystems extends beyond the Earth's physical boundaries, reaching into the very core of human health and well-being. Within our bodies, a diverse community of microbes supports digestion, immunity, and even mental health, illustrating how deeply interconnected we are with these microscopic partners. This intimate relationship invites us to reconsider notions of individuality and health, recognizing the human organism as a complex ecosystem in itself. Such awareness encourages holistic approaches to medicine and wellness that honor the delicate balance of microbial life within us. As we learn to care for these internal ecosystems, we also become more attuned to the external environments that nurture them. In embracing microbial stewardship, we embrace a future where health, environment, and science converge to promote resilience and vitality for all forms of life. Beyond their scientific and medical significance, microbial ecosystems hold a profound symbolic meaning in our evolving relationship with nature. They remind us that even the smallest forms of life have immense value, and that sustainability begins with recognizing the unseen forces that sustain us. This perspective encourages humility and patience as many microbial processes unfold slowly and subtly, requiring us to cultivate a long-term vision for planetary health. By fostering curiosity and respect for these vital communities, we cultivate a culture of care that transcends human-centered thinking, embracing the interconnectedness of all beings. Such a shift can guide policies, education, and innovation toward more thoughtful and inclusive environmental stewardship. In doing so, we plant the seeds for a future where microbial life is honored not only for its utility, but for its intrinsic role in the story of life on Earth. As we step forward from this exploration, it is important to remember that the microbial world is both ancient and ever-changing, a dynamic force that continuously shapes the conditions for life. Our growing knowledge of microbial ecosystems offers powerful tools to address global challenges such as climate change, pollution, and food security. By harnessing the potential of microbes in sustainable technologies and ecological restoration, we can foster resilience in natural systems and human societies alike. This journey of discovery invites collaboration across disciplines, cultures, and generations, united by a shared commitment to preserving the delicate balance that microbes help maintain. In embracing this partnership, we affirm our role as caretakers of a living planet, mindful of the invisible yet vital threads that weave the fabric of life. Ultimately, embracing the microbial world is an invitation to live with greater awareness and reverence for the complexity and beauty of life's foundation. It calls us to nurture not only the visible landscapes around us, but also the invisible microbial realms that sustain them. By appreciating the profound impact of microbial ecosystems, we open ourselves to a more compassionate and sustainable future, one where human progress harmonizes with the natural rhythms of the earth. Let this understanding inspire us to act gently, listen deeply, and protect diligently, ensuring that the legacy of microbial life continues to flourish alongside our own. In this shared journey, we find hope, connection, and the enduring promise of a thriving world for generations to come. And so, with quiet gratitude, we embrace the microbial world as a vital partner in the unfolding story of life,